I'm going to be walking through the basics of active transport using some diagrams. So we have active transport, which is the movement of a substance against their concentration gradient going from low to high concentration. Because it goes against the natural flow, it requires energy, which is usually ATP. So firstly, we have primary active transport, which directly uses ATP to pump a molecule across the membrane. In the diagrams I've drawn, you can see the pump is in the cell membrane. This pump uses ATP to move ions like sodium and potassium, which I've drawn in pink and red, against their gradients. ATP is used to change the shape of the pump so the ions can be pushed to the other side of the membrane. So next we will have secondary active transport, which is a little different. Instead of using ATP directly, it uses stored energy in the concentration gradients, usually created by the primary active transport. As you can see in the diagram, the sodium ions move down their concentration gradient. This provides energy to bring other molecules like glucose at the same time. This is why it's called secondary. It relies on a gradient that is originally created using ATP, but it doesn't use it directly. So active transport also includes vesicle-based processes like endocytosis, which uses ATP to bring large particles or fluid into the cells. There are three types that I have drawn out. So there's phagocytosis, which is cell eating. This brings large particles into the cell like leukocytes. We also use phagocytosis to engulf things like bacteria. Next, there's pinocytosis, which is cell drinking. Here, the cell takes in fluid containing dissolved substances. You can see the membrane forms a little small pocket that pinches inward to bring this fluid inside. A way I have to remember this is Pinot, like Pinot Grigio, is a drink. And lastly, we have cell-mediated endocytosis. This method is highly specific. Receptors on the cell will bind to certain molecules, and only when they are matched does the membrane fold inward to bring the substance inside the cell. So lastly, we just have exocytosis, which moves material out of the cell using vesicles, so opposite of endocytosis. A vesicle inside the cell will fuse to the membrane and release its content into the extracellular space. This is important for things like waste removal. To summarize everything, active transport uses ATP to move substances against their concentration gradient. Primary transport uses ATP directly. Secondary transport uses stored gradient energy. Endocytosis brings material into the cell. Exocytosis brings material out of the cell. I hope that was all very helpful to explain active transport. And then I'll just go into my study techniques. So first, I use whiteboards a lot. I like to write down any important notes or key terms, and I also like to draw diagrams and label them and connect everything together, just like I did here. And then I also use an app called Gizmo. This app is an AI-generated app. It makes flashcards and quizzes just off your notes. Just import them in. So I use this for our key terms that we have. I will put them in there, and it helps me study for them and understand them a lot better. And I hope that is helpful for your study.